So hello everyone, my name is Wolfgang Hankeln and I'm sitting here with Neil Davies. We are at the GSC14, and first of all, could you please introduce yourself and tell us what your fields of research are?
Yeah, my name is Neil Davies. I'm the director of University of California, Berkeley's research station on the island of Moorea in French Polynesia, the GUMP South Pacific Research Station.
Okay, so have you been to the GSC before, or is this the first GSC meeting that you attend, and what were the themes that are most interesting to you?
No, I've been before. I think GSC11 was the first one, so we're at 14 now. So I've been to 3. The first one was in Cambridge, so another one in England. And since then I've joined the board of the GSC also, so I'm very much involved. After the first meeting I went to, I became very active within the GSC.
And what are your main topics that you are interested in working with the GSC?
Yeah, well, I guess when I went to that first meeting, it was because I had an interest in biodiversity genomics, and as it happened, that was an area that the GSC was really ramping up in, and so it's been a very natural collaboration.
So what did you enjoy most so far at this meeting? It's the second day now.
Mm-hmm. I think this meeting's been really interesting because there's such a diverse range of sites, places in the world that are really intensively monitoring their environment using genetic techniques, and also different projects and organizations that are institutions trying to promote genomic approaches. And so just the rich range of people, but all kind of coalescing around, we need to understand these places in the world. And of course I have interest in my place. So to hear what others are doing at their different sites, how they're innovating, how they're overcoming some of the challenges that I recognize on our own site is incredibly exciting and informative.
So what do you think is the main contributions that the GSC offers to a wider community? Well,
I think obviously there's standards. I recognize that just to kind of connect up all of the research that goes on at my particular site, we need some standards across those projects. Now, if I had to tell people that they're going to come to my site, do research, they must follow some standard that I've implemented, no one would come. So it's not a problem we could solve locally, but as globally, there's a set of standards that people are beginning to implement and use. Then, when people are coming to my site, I can encourage them to use these standards which are implemented globally, and that helps us to link the data that we're collecting from our site, first of all for our own use, but it also, the GSC then makes those data standards to those scientists who work at our site and other sites. They don't have to conform to multiple different standards when they go and work somewhere else.
Can you tell us a bit more about your site? What is special about it, and also what are you investigating? What are the main focuses of your research at your site?
Right. Well, we're located on a tropical island really in the heart of the Pacific Ocean, so we're very far from land. A relatively small island surrounded by coral reefs, population about 17,000 people. So what we're basically trying to do is to understand this is a sort of microcosm of the Earth. The Earth's also an island in space. We're a small island in the middle of the ocean. We're trying to understand how that island functions. There's obviously a lot of biodiversity. There are human population living, farming, fishing, carrying out some light industry, carrying out the kinds of activities that humans do and trying to improve their lives. But very much so, because we're on a small island, it's obvious to the population that we need to preserve the resources of that island to sustain future generations. So it's a very compelling microcosm. It's small enough that we can start to understand all aspects of the system. So one of the main projects that we've been doing is first of all describe all the biodiversity. At the species level, we've started to integrate genetic tools into that. So because of the scale of the system, we have the hope of having a pretty comprehensive view of the component parts of that system. And then, of course, it's never complete, but we'll have a larger proportion of the component parts described, and then we look at how those component parts interact to provide the services that society depends upon.
So you're also part of this genomic observatory initiative, and maybe can you first of all describe what is a genomic observatory?
Right. I think in the simplest terms, it's making genomic observations, so reading DNA, DNA sequencing, in a sustained way, so over a period of time at a given place. Ultimately, that is what the basic level is what a genomic observatory is.
And what is your work at your genomic observatory site?
Yeah, so we started off trying to sequence one gene from every species on the whole island. That was the Maria Biocode project. We've done a lot of species now. We're up to about 10,000 species maybe, from the coral reefs to the mountain tops. So the next phase, that was really an inventory. That's just what's there at this 3 or 4 year period of time. Now we've got part of that library built. We want to see how it changes over time. What's there next year? Is it the same? Is it different? What abundance of each species? We know the species is there, at least 1 or 2 individuals.
Mm-hmm.
How many of them, and how does that change over time? So that's where we get into the observatory part. So we're pivoting from inventory to observatory, and we're using genetic tools to look at how many species are there, but also the genes themselves. How do the different types of gene increase or decrease in frequency over time?
Now, besides diversity, what are the questions that are particularly interesting to you?
Well, the diversity is linked to functioning of the ecosystem, because when we say biodiversity, for example, at the species level, that means we have fish. You know, if you have herbivorous fish, they're eating algae on the coral reef. If you have carnivorous fish, they're eating other fish, perhaps. So by knowing what kinds of species you have there, you have some assessment of the role and function in the ecosystem. And in a coral reef, one of the things we've been learning from all the ecological work that's going on in the system is that, for example, this aspect of biodiversity that includes herbivorous fish is absolutely vital if you want to keep your coral reef a coral reef. Yeah. Because if you lose those fish, you can algae can start to dominate the reef, and you end up not with a coral reef but with an algal reef. Now that may be a good thing in some ways if you like algae, but for many of the ecosystem services which our island depends upon right now, it's based on the idea that we have a coral reef and not an algal reef. So understanding how this biodiversity changes over time has a very direct economic and social impact on the well-being of the island.
Now, having a look at it from a global scale, why do you think does it make sense to start this initiative in a community effort and around the globe at very different sites?
Yeah, I think it does, because one thing our focus is understanding our particular site. We're interested in comparing what we see at our site with other places. But genomic technologies in particular are advancing so rapidly. They seem to offer such great potential, but there are some enormous challenges, technological, the kinds of equipment you need, the kinds of processing you need to do on the samples, and how you treat the data. And those such a fast advancing field that we don't want to make all of the mistakes ourselves. We'd rather profit from the mistakes that others have made and share with others the mistakes that we've made, so that we can best apply these new technologies and these new approaches the most efficiently as possible. And then for that, sharing across the network is really a very powerful approach.
Now, compiling this huge global set of data, what do you think are the hopes concerning the questions that can be addressed with this comprehensive set of data that?
Right. Yeah. Well, science depends on observations of the natural world. Mm-hmm. We could make up things in our head, and they may be very interesting, but they may have no relevance to what actually is happening out there in the real world. So we need observations of the real world. Genomic technologies give us a new lens of which we can see things we could never see before. So that's a great opportunity. It generates a lot more capacity for observation, gives us a different view of the natural world, so that can help us generate our explanations of how that world works with more confidence, and maybe come up with new theories to explain these new data that we're seeing. And maybe these new data that we see will help us to answer some of the older questions that we haven't been able to address properly yet. So those are the big advances that these data sets can provide. But again, one of the interesting things about this meeting is we've got one session that's specifically looking at, okay, with all these new data and these new technologies, of what is the theory that enables you to profit from those new data, and what are the gaps in theory that no one had to come up with a theory before because no one had those data before. So, I mean, obviously, before we knew anything about microbes, there was no theory about infectious disease and how to manage it based on, because no one knew microbes were causing the infectious disease. So the new data, new observations lead to new and require new theoretical advances.
Okay, now talking about the infrastructure aspect. So there are already existing infrastructures, and how is the GOES initiative planning to get integrated into already existing infrastructures?
Right. Yeah, so that's obviously another advantage of being part of a network is you can you can utilize common resources, and you don't have to necessarily pay the full cost of building something that can be shared across a network. So that will be true within the GOES network, for example, some of the sequencing centers and some of the computational resources that are available at centers around the world. Different sites can take advantage of those. And also as a network, the Genomic Observatories network itself can take advantage of other networks which are addressing ecosystems and other systems at different levels. For example, the ecological research networks. They're not necessarily using genomics, although genomics can contribute to ecology, but they're providing a lot of the contextual information. The same thing with the environmental research networks and the satellites, into which we put the genomic layer. The genomic data stream fits in and leverages the ecological data streams and the environmental data streams, and ultimately the social and economic data too. There's a lot of fisheries data, agricultural data, economic data that we want to know how if biodiversity is changing, what impact is that having on people's lives.
Why is your Genomic Observatory site also participating in the OSD initiative?
I think OSD was a great opportunity and perhaps by no coincidence came at the same time as the Genomic Observatories network was getting off the ground. To me, it provides a common demonstration project. It's a project that we can all participate in. It's relatively low investment in the sense it's one day, so we can do that. We can conform to whatever standards the group of sites and microB3 and others agree on. We can contribute to that. We can see how it works, how it doesn't work, and the data set that we generate together. We can see how powerful that is because we follow these common approaches. If we're able to do things with that relatively small data set that we couldn't do with the rest of the data that we're collecting, that may motivate us to say, oh, well, actually, if we started applying some of these approaches to everything else we do, then we'd be able to share things with these other sites. We'd be able to ask these new questions that we can't ask if we stick with maybe the systems that we're currently using.
So in terms of this data set and the power that you have mentioned, what do you think are kind of the low-hanging fruits or the easy scientific questions that you maybe can answer compiling this data set at the OSD?
I think some of the questions are the traditional questions that we've always had, those of us interested in biodiversity and studying it. For example, things like the species-area relationship. The bigger island you have, the more species you have, or the latitudinal gradients in biodiversity that we see. More species at the tropics, fewer at the polar zones, and in the Pacific we have a biodiversity gradient that also goes from Australia, Southeast Asia, which are very high in diversity. As you go out across the Pacific towards Tahiti, you gradually lose biodiversity, so we're at a low end of that gradient. So those gradients and relationships are well known at the species level, from collecting butterflies or counting plants or what have you. Do they apply at the microbial level or at the genetic level? There's some evidence on that, but the challenge there is collecting data that are comparable. Yeah. So this is one potential way with OSD. We can start to get data sets that are comparable, and we can start to address, well, do we see the same kinds of pattern at the genomic and microbial and other kinds of taxa?
Would you agree that this OSD initiative also opens a window on monitoring human impact on the environment?
Yeah, that I think is, in terms of applications, is one of the most exciting things about genomics. Is, of course, it's new. Until now, it's been incredibly expensive and difficult and hard to do. But the costs are falling so rapidly that soon it's arguably going to be easier to read DNA than to count fish. And, it may be the best way to monitor change we still have to do some sampling, but the sampling can be done, DNA can be read, DNA is very consistent standard. We can compare it. We can look at how it changes over time at our site, compare it with other sites. So it may be that these tools enable us this window on biodiversity that can very efficiently generate streams of data that give us these kinds of comparative capacity to monitor change. And that's when you look, once you've studied something long enough, we may see that certain DNA signatures are associated with certain events that we want to either avoid or we want to know about them in advance. For example, it could be an outbreak of an infectious disease to the human population directly or impacting a coral reef or an agricultural system. And if we could see that ahead of time and prepare for it and maybe avoid it or at least adapt to it, some of these genomic surveillance technologies may be the most efficient way to do that.
So thanks a lot for taking your time and answering the questions patiently, and have a nice rest of the meeting.
No, you're welcome. Thank you.